Hello, everyone, and welcome to today's webinar, Advancing Oligo Therapeutics Drug Development. This webinar is part of the 12th Annual Precision Medicine, Genomics, Genetics, and Molecular Diagnostics Virtual Event Series 2024. I am Israel Gonzalez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, visit www.thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find this live chat feature located to the left of your screen. You'll also notice that the speaker has provided a survey located on the left of the screen. This survey will be available during both the live session and the on-demand period. In addition to asking questions, we also encourage you to participate in 3 poll questions at the beginning, middle, and end of the speaker presentation. Click on your answer when the poll questions are displayed. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now let's get started. I would like to present today's speaker, Gary Held, Staff Scientist, Analytical Development, Nucleic Acid Therapeutics, Thermo Fisher Scientific. For a complete biography, please visit the Presenter tab from the menu on the left of your screen. Gary, you may now begin your presentation.
So it's my pleasure to be with you here today. My goal is to help give you insight into considerations you should have when choosing starting materials for your oligonucleotide therapeutic, and that specifically meaning the phosphoramidites that are used in the synthesis. So my experience has been on the quality side of manufacturing and testing of phosphoramidites for over 18 years, and I currently work at the Thermo Fisher site in Milwaukee, Wisconsin, which we consider ourselves a center of excellence for nucleic acid chemistry within Thermo Fisher. So I remember when I first started many years ago, the overall purity for phosphoramidite specifications was just HPLC and NMR. As we'll talk about today, there are many types of phosphoramidites used in therapeutics that weren't before, and the chemistry of them is more complex, and the technology to test them has become more mainstream and easier to use. And that specifications have also evolved to ensure consistent product quality and more complete understanding of impurities in their formation. So over time, the experience of a robust supply chain and supported by a consistent, scalable supply of product will improve the overall product qualities. So today I've outlined the topics I'd like to cover. So we'd like to talk about the criteria for choosing any raw materials in the development of a therapeutic based on the stage of the drug development, and each has unique challenges and requirements. Then specific considerations are needed in picking amidites that will go into the synthesis. Next, we'll look at the synthesis of phosphoramidites and the areas that have potential for introducing impurities into that final product. And then we'll talk about how these impurities are classified and what mitigations you need to consider and what type of specifications are meaningful to your product. Next, we'll dive specifically into the analysis of one amidite, that being 2'-fluorophosphoramidite, to show the types of variation that can be expected in a vendor-to-vendor analysis, and then look closely into the real impurities present in this amidite produced today. And lastly, I want to take a thoughtful look at a potential solution to all the product purity and impurity considerations we'll talk about, that being the TheraPure line of phosphoramidite products, all in an effort to make your oligotherapeutic a success. So when we think about the overall drug development process, taking the oligo through this path, we find several key decision points. The drug discovery, in drug discovery, the goals are around validating the drug target and drug candidate. And then as we move into the drug development, demonstration of the drug in humans, the regulations increase. Controls are added to the production process. More specifications are needed in the process, as well as in the raw materials. Larger quantities of materials are needed to do the studies and trials that are necessary, And then finally, full into drug manufacturing at scale, and the ability to make changes severely decreases. The consistency of the process and raw material supply is essential. Understanding the process and the materials that go into this process are critical as well. And the risk of raw materials needs to be minimized. So using a supplier whose material has been used in the development process before and has consistent quality and the ability to deliver at scale is key.
We would appreciate your participation by answering a few polling questions. To record your answers, click on the option on the screen. Our first polling question is, at what stage of mRNA or oligo therapeutic production process are you working at? A, have not started yet. B, early process development. C, late process development. Or D, have an established process already. I will give you a few moments to select your answers.
So let's talk about what your key considerations should be when looking at your raw materials. The number one is quality. So we'll look at purity and discuss more about the impurity profiles and things to consider. So what about product stability? What are your needs for storage conditions of the product to maintain that quality? Can your supplier provide you the information you need about this stability? To go along with stability is the packaging that is needed for your phosphoramidite. Will it be a customized package? Can the package be scaled to the size and number of pieces that you need? Are precautions taken to protect the environmental conditions such as light and moisture with amber bottles and argon purging? Do you see needing a custom packaging system? Can a customized package lower the overall handling of your product at your site or within your synthesis process? All are considerations for your raw material delivery. Consistency. Consistency is an important aspect. Can the supplier control batch-to-batch variation in the impurity profile? Is it produced with consistent overall purity? Does the material have consistent physical characteristics? Are appropriate in-process controls monitored to achieve this? Do they monitor and qualify the materials that go into their production? Proven products. This really comes down to experience. Has the material that you are using been used in other successful drug candidates? Is there a track record of taking a small scale process and bringing it to full routine batch scale? If so, has the quality been maintained? Things to consider. We move to support. When considering the raw materials, do you have easy access to the product support and quality resources you need to complete your project? Are you able to get into direct communication with scientists like me, manufacturing personnel, and customer service to meet your needs? Do you get the documentation support and communications you need to be successful? And then finally, scalability. Have these raw materials been made at sufficient scale with large enough levels of inputs that will meet not only your current demands, but the demands of your product at launch? So now if we dive a little bit more into talking about the phosphoramidite molecules themselves. So phosphoramidites are the building blocks for synthesis of oligonucleotides. So their core structure is a base that typically has a protecting group and is linked to a sugar. Then two additional functional groups are added to aid in the synthetic synthesis. So we have the dimethoxytridyl group at the 5' position, and then reacted with phosphorylating reagent in the 3' position to make it an amidite. The simplest form of these being really the DNA phosphoramidites shown here, followed by the now common 2' modifications of RNA phosphoramidites, whether that's 2' methyls, 2' Mos, or 2' fluoro modifications, and additionally any conjugates that can be added to the oligo in the form of GalNAc as an example for delivery. So to make an oligo, we take these individual phosphoramidite monomers and use a solid support system, react them through this iterative process on the right. First, detrotylation, in which the dimethoxytrityl group is removed from the 5' end. This is where the amidite solution is then introduced with an activator and is added as a single unit to the growing oligo chain. So the next step is the oxidation of the phosphotriester linkage and converted from a phosphorus in the P3 oxidation state to a phosphorus in the P5 oxidation state. Then we move back over to capping, where any remaining hydroxyls are capped, and then the process starts over again, growing the chain. The next amidite is introduced, and the cycle continues. As each base is added in the cycle, there's a potential for an impurity inherent in that phosphoramidite to be added as well. But not all amidite impurities are created equal. Impurities are defined by their relative inability to be removed in the manufacturing process. So what type of impurities are there? And I've listed them here. They fall into two main categories, the critical and non-critical. Critical impurities are ones that allow for participation in the synthetic synthesis that may cause things like branching or deletion or reverse bases. They contain amidite functionality, and they may or may not have the ability to be removed in the overall oligosynthetic process. Non-critical impurities can be of two types: ones that do not participate in the synthesis at all and are typically in the P5 oxidation state already, versus the reactive P3 oxidation state. They do not react or participate in the coupling reaction at all and most likely are purged out later in the process. The other is reactive, non-critical impurities, because they still may react within that coupling cycle, but still make the full correct.
Our next question is, would you like to be contacted by a technical representative to discuss about the raw materials for mRNA or oligos therapeutic production or manufacturing process? A, yes, or B, no. Please take a few moments to select your answer.
So we want to touch a moment on how amidites are synthesized in order to understand the impurity formation, how impurity formation can occur. So if we start in the top left there and look at DNA phosphoramidites, these are the most widely used building blocks and provide the most basic understanding of the impurity formation. So if we start up in the top left, we have the bases A, C, or G, and then we add base protection to the free amine. So for A and C, we'll add either benzoyl, or in the case of G, isobutyryl group as the protecting group. So this is the first major place in which impurities can be introduced. And the main mitigation step here is to use high quality protecting group reagents or purification at this step if needed. Thymidine does not have a free amine, so therefore we skip this step and move on to the next step, which is the addition of the dimethoxytrityl chloride at the 5' position. So we'll talk in a minute about some of the common impurities in the DMT group and the path that can lead to critical or non-critical impurity. And then finally, the last step is the phosphorylating reaction, in which the addition of the phos reagent in the presence of an activator adds at the 3' position to complete the amidite synthesis. The addition of these reagents has the potential to a number of different types of impurities. So strict in-process controls and raw material specifications are key to minimize the impurities at these steps. So here I've highlighted in the red boxes the main areas of the molecules that have opportunities for impurity formation. So if we break this down a little bit more and look at the critical and non-critical impurities and their classification. So if we start with critical impurity number 16, this would be reactive in the oligosynthesis cycle and produce a parent oligo with a reverse linkage on it. So it has 3', 5' are switched. This impurity in the oligo chain will most likely not be purged during the downstream processing and hence be critical, as it would be incorporated into the final product. The next two variations of non-critical impurities are number 17 and 18. Number 17 is a reactive and non-critical impurity. In this example, the DMT chloride reagent contained an impurity with one chlorine that's been added to a ring. It is expected that this would participate similarly to the normal DMT in the oligosynthesis, and the DMT plus chloride would be cleaved and removed from the oligonucleotide in the normal detritialization process, resulting in the expected full-length product. Next, we have compound 18, which is a non-reactive, non-critical impurity in which the phosphorus is already in the P5 oxidation state. So it wouldn't participate at all in the synthetic coupling reaction and would be purged out of the process. And then finally, compound 19, its criticality may not always be clearly defined. So this compound is missing the base protection. Whether it was never installed or it was cleaved at some point, it would be reactive but cause branching that may or may not be removed later. But at the same time, it could be considered a non-critical impurity if it was the last base added in the oligo process. This would still lead to the correct full-length product. So specifications for phosphoramidites are set with this understanding and terminology of impurities, and suppliers should understand what impurities make them critical or not, and minimize them in their process. So as we talked about before, a source of introducing an impurity into the phosphoramidite can come through the addition of the dimethoxytrityl group. This example shows the importance of minimizing upstream impurities and controlling the supply chain and understanding the testing materials before they are used. So in this example, we're adding dimethoxytrityl chloride to a protected DC. And so on the top is the normal expected reaction with the base protected nucleoside reacting with the DMT chloride to produce the expected product. And then upon reaction with phos reagent gives the correct final amidite. In the middle reaction, would be a modified DMT containing acetoxy group, or it could be chlorine as we previously saw before, but some kind of modification to DMT that would then react with the phos reagent and create this non-critical impurity, in which upon detritialization would be removed from the amidite and oligo process. And then finally, if we move along the bottom, in which the dimethoxytrityl is missing a methyl group, and it gives us that free OH, that could be phosphorylated similar to the 3' position and be reactive in the oligosynthesis and therefore be considered a critical impurity. So this could be an example of how changes within the supply chain can add new impurities. So our understanding and following these impurities to their source and then adding the relevant specifications to control them is really key to overall amidite purity. So if we take a look at other types of amidites, in this case 2' modifications such as 2' Mo, this synthetic step makes this material more complex as well, and with the addition of a new functional group in this 4th area on the 2' position. So if we take a look at what 2' Mo impurities to expect, common 2' modification impurities of Mo would be the ones listed as R groups here, and these impurities may arise as a result of the chemical synthesis, maybe alcoholic solvents present, or some type of impurity in the Mo reagent itself. Additionally, a 2' modification now allows for additional regio-specific impurities to be possible. So in the case of impurity 36 listed here, the 3' 5' switch can occur similar to what we had seen in DNA, but now we have the possibility of a 2' 3' switch as well. Both of these regio-specific impurities would be considered critical, as their incorporation into the oligo would most likely not be purged in the downstream processing. And then just to finish out the 2' modifications, we have some, initial unique impurities that can form in these other 2' modifications of fluoro and O-methyl, or even the more complicated synthesis of making LNAs or CETs with their additional lengthy synthetic process can also introduce additional impurities to be wary of. So now we move on from the chemical synthesis itself and take a look at a case study in which we did an analysis of the 2'-fluoro-A amidite. And so we took a look at the impurity profile to show vendor-to-vendor variation. We took a look at 4 different vendors. From this, we did LCMS analysis with high-resolution accurate mass, and then tried to identify all impurities at near the method detection limit of 0.01%. So this is the UV analysis of the 2'-fluoro-A amidite from 4 different vendors. So first thing you notice is the number of impurities present, and the HPLC separation can be fairly complex, and we typically see maybe 10 to 25 to 30 impurities present. Similar to the main amidite, these impurities also have multiple isomers as well to be aware of. So I want to point out a few differences here in these 4 lots analyzed to show the variation of the impurities and the impurity profile from different vendors. So if we take a look at the difference at about 7 minutes with peaks 5 and 6 being seen in vendor A lot only and not present in the other ones. And then we'll also take a closer look at peaks 11 and 13, which are very low level impurities mixed in with some other impurities. So we'll try to take a look at that more closely. And then we see quite a bit of complexity around peaks 21 through 24. So we'll use the mass spec to take a look at those as well. So let's begin by looking at the mass information for peaks 5 and 6 that we saw in the previous slide that were unique to vendor A. So we find the mass of these 2 impurities is really the same. We see 677 or 678 as a M+1 impurity. Having these 2 peaks of the same mass is really a tip to us that they're most likely just isomers of the same impurity. So we have this proposed structure for the impurity in which the 5' is not DMT but is benzoyl, here shown in the blue. And from the knowledge of the synthetic process, it's really a very reasonable structure to propose. And now that we can use MS data to confirm this, we further look at the fragmentation pattern of the parent ion of the 678, and it forms into the expected ions of the proposed structure. So the observation fits the proposed structure, and we have determined that these impurities are non-critical, so no TMT to deprotect, and then we most likely purged.
Our final question is, what team are you in? A, discovery, or B, development? Thank you so much for participating in these polling questions.
We move to peaks 11 and 13 we saw before. These peaks are very small and close to the limit of quantitation. But we are still able to determine that each peak is actually a coelution of 2 other impurities, distinguishable by mass spec. So we found 2 different masses present, 774 and 772. And using the same process as before to determine the proposed structure, the 2 impurities are proposed, which again makes sense with what we know about the amidite and how it's made. 774 being bis amidite, and 772 missing the base protection. And so one thing of note here is the relative amounts of each can be shown with the extracted mass below. So note the difference in the relative scale. These 2 impurities, it's 772 extracted ion being 10 times lower than that of 774 extracted. The different colored chromatogram traces show the different amounts in each of the vendors from the previous slide. So using UV and mass spec is a very powerful combination to really help us with the complexity of the separation and the types of impurities that are present. And then finally, one more example, when we focused in on the 10 to 11 minute range of the chromatogram, using the mass spec, we determined that this group of peaks 21 through 24 all had the same exact mass of 910. So when you think about what are the possible impurities with mass 910, well, we can infer from the chemistry and what we had seen before that we'd expect this to be an amidite plus a chlorine somewhere in the molecule. So the extracted ion chromatogram of mass 910 is shown here with the differences in the number and the amount seen in each vendor's material. So we're able to next go to figure out specifically which peak is which. Again, using MS/MS, if we look at the two peaks, specifically peak 21 and peak 24, we see that both have masses of 910, which we expected. But then when we look at the fragmentation pattern of these two is different. In the MS/MS of peak 21, we see a fragment 303. So 303 is a classic fragment we typically see for all amidites for the unmodified DMT group. So for peak 21, we would not expect the chlorine to be on the DMT group. Instead, the fragmentation shows a peak of 274, which is indication that the chlorine is on the benzoyl group. Now moving to the peak 24 MS/MS data, we don't see the 303 peak, but we see a 337 peak. So this 337 peak would be for a DMT that has added chlorine somewhere within the ring system. This is a very typical impurity in amidites, and so applying the same technique to the other 910 that we saw. So putting this all together, we're able to make an individual assessment to each peak. Why within this chromatogram, whether the chloro is on the benzoyl or the chloro is on the DMT. But then why do we see multiple peaks? Well, remember that the combination of amidite has typically two isomers, and then the potential positional isomers of chlorine within the ring system itself. So the good news is all these peaks would be considered non-critical, as they would be reactive upon their loss of the, they're non-critical, they are reactive, but upon the synthetic process, either the DMT group is lost or the base protection is lost, and would result in the correct full-length product. So finally, if we put all this information together about all the impurities using the UV HPLC with the help of a high-resolution mass spec, and along with the knowledge of the chemical synthesis, we can put together a clear understanding of the impurity profile of this one amidite. So shown is just a list of peaks that were actually detected in these amidites and a proposed structure for each of these. So you can imagine an even more exhaustive list of other potential impurities, but typically this is a very typical pattern of impurities present in amidites. The diversity of impurity types also shown, which each functional area of the molecule that we talked about has some type of impurity or modification, whether that be the 5' DMT, somewhere on the base protection, in the 2' modification, or the 3' amidite position, also have potential to introduce impurities. And so this table has assigned an example classification for each impurity, as it would be critical or non-critical. So this is where your understanding of your own oligosynthesis is important in making this distinction of impurity types. Many of these impurities are well understood and can have a criticality assigned to them, but the level that your process can tolerate really is key to the overall determination. So I've shown you what considerations you should have in determining the quality of amidites used in the process. So considerations around impurities, particularly, but overall quality, consistency, and scalability as well. So one solution to this could be looking at a supplier that has these considerations already in mind. So I want to share with you the one in the form of the TheraPure phosphoramidite product line from Thermo Fisher Scientific. So this portfolio of offerings includes all the main modifications we've talked about today, and also a variety of additional base protecting groups depending on your oligosynthesis needs. So these include the DNA, the RNA, the 2' O modifications of O-methyls, fluoros, and MOs, as well as the conformationally restricted LNA amidites. And the Therapure products already take into consideration the types of impurities we talked about today, having specifications for both the critical and non-critical impurities. And these impurities are limited by building the quality into the process, controlling and monitoring the supply chain of raw materials, setting in-process controls, and then finally testing them to a meaningful set of release specifications. The analytical techniques used in the final testing, release testing, include not only HPLC purity supported by the mass spec that we talked about today, but also include NMR. So phosphorus NMR gives us an orthogonal method to confirm purity, but looking for phosphorus-related impurities that may not be UV active. So headspace GC is used to determine the levels of residual solvent in the product. And then finally, the oven Carl Fisher is used to determine the water content of the products. So this oven technique is used on amidites, as they will react with the KF reagent directly and give the false high water results. So this is an example of a certificate of analysis for Therapure amidites. It's showing the comprehensive list of testing that's done and is included with each lot of product. So the testing also includes some visual determinations and tests of solution clarity to show performance of the material. And then you can see the purity is measured by HPLC with considerations for levels of critical and not critical impurities. And the purity is also verified with the use of the phosphorus NMR. And then water content and residual solvents are included. And finally, the products are certified to be animal origin free by diligently looking at all the materials that go into the synthesis, as well as any packaging the product may see. So we touched on these key factors to consider in your raw material choices for phosphoramidites, with number 1 being the quality, number 2 being customized packaging offerings, number 3, consistency of batches, 4th, the ease of access to regulatory support and quality team, 5, choosing a supplier with experience in taking the product from small scale to large scale to meet your drug development needs, and 6, partnering with a supplier that has proven products that have already been used in approved drug products. Using proper raw materials that meet the requirements for synthetic oligonucleotide therapeutics is really a big key to success. And finally, we touched on that not all raw materials are the same. Phosphoramidites contain a wide variety of impurities. Determining which one of these impurities may be critical or not critical in your process is an important consideration when choosing your amidite supply. So again, one solution can be the phosphoramidite, Therapure phosphoramidite materials with their well-defined manufacturing process, high quality standards that checks the boxes of extensive testing, minimized critical impurities, and consistency of product at scale. So with that, I'd like to thank you for your attention and would love to take a few questions.
Thank you, Gary, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank Thermo Fisher Scientific for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until May 15th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time.